So now that we understand the basics of the dogma of molecular biology, the mechanism, the mechanics of taking the information in DNA and making it into a protein, I want to talk about the differences between eukaryotes and prokaryotes, specifically with regard to what's called alternative splicing. So again, we know the dogma of molecular biology, DNA to RNA to protein to trait, but we have mentioned that there is a difference in transcription. When we make a copy, a photocopy of that reference book, when we make a copy of the RNA from the DNA, it's slightly different in prokaryotes than it is in eukaryotes. Again, going back to our concepts of the eukaryotic cell, why do we have different rooms in a eukaryotic cell? For optimization, to optimize the chemical activities, the biochemistry in that room. Well, prokaryotic cells don't have a nucleus. So this would suggest that there's something going on in the nucleus that prokaryotic cells don't need to do. And there is. In this case, it's the processing of a pre-mRNA to an RNA molecule, the final mRNA. So what we're going to see in eukaryotes is that we're going to make an exact copy of the DNA, and then we're going to change that copy, that was called the primary transcript, before we send it out to be translated. And so this seems like a lot of extra work. Why would you do that? Because what we're going to see is that there are sections in genes that don't encode for protein. And these are called introns. So you'll see in this picture the RNA transcript, the pre-mRNA has those little light pink loops in it that say intron versus the exon. Introns don't encode for amino acids, but the exons do. So you actually have to snip or splice out all the introns and then put all the exons back together before you send it out. Otherwise, you get a faulty protein. Now, this seems like a whole lot of extra work. What is the point? Why do eukaryotes, being more complex, have to make this more complex when it seems like a wasted step? Well, let's talk about the structures of genomes and genes just a little bit more. So again, a genome is all the DNA that you have. And the human genome is about 3.1 billion bases. Right? Now, there's a lot to be said about this topic, but we're going to keep it simple for now. Of all of that DNA, only about 2%, 1.5%, actually encodes for protein. Whoa. Why would you keep all that DNA and not have most of it encode for protein? Now, the other thing to note is that the introns, these sequences actually inside genes that don't encode for protein, are quite a huge part of that genome. Now, hold on to that idea, and then let's look at prokaryotes. Because remember, prokaryotes don't have introns. What you see is what you get in a prokaryotic gene structure. It makes an exact copy of the DNA, and that is ready to be made into a protein. This also lends itself to the speed at which these prokaryotes are able to respond and make protein. Now, look at this chart. This chart says the percent of DNA not coding for protein. And you can see that increases significantly as we go from prokaryotes, the simplest of cells, to single-celled eukaryotes, to more and more complex, quote-unquote, animals and plants, with humans at a significant portion of our genome non-coding sequence. All right. Now, the other thing to think about here is the fact that when we first started studying the human genome, we thought there would be probably somewhere between 200 or 300,000 genes, because we're pretty complex. And we suggested that number because prokaryotes, specifically E. coli, which is a bacterium that's very common in your intestines, has about 5,000 genes. So we said, wow, 5,000 genes, we're much, much more complicated than a prokaryote. We must have at least, 200,000 or 300,000 genes, if not more, because we're so complex. What we found out as we sequenced the human genome is we have about 20 or 25,000 genes. And people were really taken aback by this. They said, wow, look how much more complicated we are than this single-cell prokaryote, yet we only have 5 times the number of genes? That doesn't make any sense. But when you think about the structure of our genes, the structure of these genes with introns and exons, whereas prokaryotes, E. coli, doesn't have that, all of a sudden it makes more sense. So let's look at eukaryotic DNA one more time. Here's that gene in your DNA with the introns in it. And remember, we have to go through this RNA processing. We're going to make an exact copy, this primary transcript, this precursor mRNA that has the introns and exons all lined up just like the DNA. And then we go through processing, the RNA splicing. We're going to remove these yellow segments and put all the pink segments together. And that is the mRNA, the messenger RNA that will travel out to make a protein. So you say, well, why do we do this? How does this work? Well, the beauty of having those exons is that they allow for what's called alternative splicing. That I don't have to just remove all the exons the same way every time. So what that does effectively is say I can have one gene in my DNA that I can make many different proteins from based on the way I splice the mRNA. So here we can see the pre-mRNA with all the exons labeled 1, 2, 3, 4. Okay, remember the exons are the part that encode for the genes. And I can put them together into an mRNA differently. So I may put all the exons, splice out the introns to make protein A, which is just exons 1, 2, and 3. Or I may splice out the exons in a way that removes the introns out in a way that removes the exon 3 with them. And I get protein B, which is exons 1, 2, and 4. So what we found is that on average, every human gene can make 8.7 different proteins. 8 times 25,000 is 200,000. Now we're back in our ballpark for complexity. So the proteins that we see, the different proteins in the cell, actually may be coming from the same gene based on the way they're spliced. General trend here, again, eukaryotic life tends to be more complex than prokaryotic life, all the way down to their gene structure. Genes in eukaryotes have introns and exons. The exons are the portion of the gene that code for amino acids that will ultimately become a protein, and the introns are non-coding segments that need to be removed from the copy that we make of RNA to make the final RNA that will then be made into a protein. We can make that final mRNA differently through alternative splicing.